Invitation to present. It's great to be here this morning. So yes, I will tell you this morning about work that we've done understanding immune responses to cancer from single cell data, and particularly focused on systemic immune responses, what's happening outside of the tumor broadly across the organism. And so just as a bit of a backdrop, in my group, we're really interested in how the immune system makes decisions. This is the process that we find really fascinating. And we think about it at multiple scales of organization. At the broadest level, organismally, we know that the immune system is responsible for regulating lots of physiological processes across the organism. And these different physiological processes have to be coordinated with one another. Have a little bit of background noise, so if you're just joining, maybe check and mute yourself. At the tissue level, we think of the immune system as an organ without borders. So immune cells constitutively infiltrate every tissue across the organism, pretty much. And despite the fact that cells are located at really large distances away from each other, they somehow still need to coordinate their behavior to form coherent immune responses across sites. And then at the cellular level, we know that the immune system is comprised of lots of different types of cells that have unique identities and roles and responsibilities. And again, somehow these cells need to communicate with each other to coordinate responses. And particularly at the cellular level, we find it particularly fascinating that there is no master regulator cell of the immune system. There's no single cell that provides cues and directs the behavior of every other cell type, but rather this is really an instance of collective decision making. Essentially, every cell type produces factors or expresses ligands that regulate the activity and function of other immune cell types in the system. And so how this sort of collective decision making takes place is a really fascinating process to us. Now, I would argue that it's really this latter element, the cellular diversity of the system, of the immune system in particular, that's prompted the development of single cell technologies historically. We've known that different types of immune cells behave in different ways. And so we've really always needed to study cells at the single cell level to be able to discriminate what different cell types are doing in different contexts. And the mainstay workhorse for doing this historically has been flow cytometry. So using this technique, we start with cells that we're interested in analyzing, and we incubate those cells with antibodies that are specific to proteins that we want to quantify. And each of these antibodies is tagged with a fluorescent dye that has an excitation property and that will give off emission photons when it's excited. And so as these cells transit through a series of lasers, one cell at a time, the lasers will excite these fluorescent dyes. The fluorescent dyes will give off emission photons, and we can quantify those emission photons as a measure of protein expression in that single cell. And the way that we multiplex by flow cytometry is to use antibodies that are tagged with different fluorophores. Now, the major limitation here is that these fluorophores give off photons at a range of wavelengths, a spectrum of emission photons. And as a consequence, these overlap with one another and sometimes make it difficult to discriminate which fluorescent dye gave off a particular signal. So just as an example, if we detect an emission photon here at, say, 550 nanometers, there's some probability that that could have been given off by this blue dye or this green dye or this yellow-green dye. And we can solve this problem to a certain extent using a mathematical process called compensation to try to filter out the pairwise overlap in signal between all of these fluorophores. But every time we do that, we introduce some element of error into the data. And so there are only a certain number of fluorochromes that we can use in a traditional flow cytometry assay before it becomes really unwieldy. And for those of you who are sort of aficionados in this space, you may know there have been some recent advances in spectral flow cytometry. That's an exciting technique that allows us to do more multiplexing, but using conventional hardware, generally measuring somewhere between 10 and 15 proteins at a time using fluorescence-based flow cytometry. And it can take quite a long time to optimize antibody panels at that sort of dimensionality. And so a technique that we started working on when I was a graduate student at this point, about 10 years ago, is one called mass cytometry, which solves some of these problems. And so using mass cytometry, instead of tagging antibodies with fluorescent dyes, we instead tag antibodies with purified heavy metal isotopes. And the advantage of doing that is that we can then use a mass spectrometer to quantify the abundance of metal on each individual cell. And as you can see, the detection peaks for each element that we tag antibodies with are very well behaved. There's very little spillover or bleed from one parameter into another parameter using mass cytometry, or CyTOF for short, as it's called. And so this is an exciting technique that we still use quite a lot in our lab, and really, I'd say, opened up a lot of opportunities for starting to do what we'd call systems immunology, which is to ask questions about how the entirety of the immune system is functioning in a particular context, looking at every cell type simultaneously and asking interesting questions about how those cells are functioning. And so that's the technology that I will talk to you a bit about today. Some of the work that we've done understanding systemic immune responses to cancer. It's a great technique for these types of studies that I'll show you, because the throughput of the mass cytometry, or CyTOF, is quite high. We can easily analyze several hundred samples using this type of workflow, and the data are very highly quantitative, which gives us opportunities to use tools from machine learning and statistical inference to really identify interesting patterns in the data. And so to start, I'll tell you about a little bit about a project that we did a few years ago to lay some a backdrop here of where we've gone more recently. So this was a study that we did trying to understand what makes effective immunotherapies effective. A relatively simple question. And we started by taking mice that spontaneously developed breast cancer, and we treated them either with an ineffective therapy, which in this context was an anti-PD-1 antibody. The checkpoint inhibitors don't work in many mouse models, but this is one of them. Or we treated it with an effective therapy that we had spent some time developing that uses tumor-binding antibodies and adjuvants to activate dendritic cells. And we don't need to worry too much about the specifics of this therapy. For this talk, it will suffice to just think of it as an ineffective therapy versus an effective therapy. And the question was really, what is the effective therapy doing that the ineffective therapy is failing to do? And so to answer that question, we analyzed all of the different tissues that we thought might be relevant in mounting these immune responses: the tumor draining lymph node, the spleen, the blood, and the bone marrow. And from all of these tissues, we put cells into a single cell suspension and we analyzed them by CyTOF. And then we went about analyzing the data. And so what I'm showing you here is what we call a scaffold map, which is a graphical network representation of our CyTOF data. And I'll walk you through exactly what you're looking at here. So in this network, each node in the network, each circle represents a cluster of immune cells that was identified by taking the underlying data and subjecting it to a clustering algorithm. So asking the computer to identify groups of cells that have similar protein expression to one another. And each cluster is connected to other clusters that it's similar to with these edges. So edges representing these lines in the graph. And the shorter an edge, the more similar two clusters are to one another. Now you'll notice there are also some special nodes in the graph that are in black and that have labels. And these are what we call landmark nodes. So these help us understand exactly which element of the immune system we're looking at. And these landmark nodes are defined by our canonical understanding of immune cell types. So traditionally, the way that we analyze cytometry data is to manually gate the data, to look for combinations of proteins that we know are unique for a given cell type, and to manually identify those cells. And so that's what we've done for these landmark nodes, but we include them in this graphical network as a way to make the analysis really easily interpretable to us as the researcher. So we know, for instance, if we look over here at the right at this plasmacytoid dendritic cell landmark, we know that these clusters surrounding the PDC landmark are almost certainly different flavors of PDCs. So compared to completely unsupervised dimensionality reduction techniques like t-SNE or UMAP, this approach makes it really easy for us to immediately see which immune cell type we're looking at in different regions of the graph. And what we can do then, because we've clustered the data, is we can do things like run a significance analysis across all of the clusters and ask which of them have statistically significant changes between groups of interest. And so that's the way that we color the graph. Anything shown in red is statistically significantly higher in the mice that received the good immunotherapy, and anything in blue is statistically significantly less abundant in the mice that received the effective immunotherapy. Anything in gray doesn't have a statistically significant change. And so what we're looking at here is the abundance of all of the different immune cell types in the tumor microenvironment. And what you can see is generally a lot of red, which means that after the effective immunotherapy, many different immune cell types are at significantly higher abundances in the tumor. And that generally makes sense. We think immunotherapy is eliciting an immune response, and so there should be more immune cells in the tumor if it's working. You can see that there are some exceptions to this rule. We have some macrophages over here that are decreasing in abundance. These are generally macrophages with more of an immunosuppressive phenotype. And we also have a few clusters of dendritic cells here that are decreasing in abundance. And these are tissue-resident migratory dendritic cells that we actually see leaving the tumor early after immunotherapy. So lots of dynamic changes in terms of the cell types that are present in the tumor. Now, we can take the same analysis and we can ask different types of questions. So here what we did was to ask which cells are proliferating at different rates, expressing Ki67, which is a marker of cellular proliferation cell cycle. And this was really interesting to us because here we're looking in the tumor while the tumors are being rejected, while there's a good immune response being mounted against the tumor. And what we saw is there was no change in immune cell proliferation between mice that received the effective therapy compared to those that received the ineffective therapy. Now, this was surprising to us because we know that good immune responses are associated with clonal expansion of antigen-specific lymphocytes, generally proliferation of lots of immune cells. And we weren't seeing that in the tumor, which was a bit puzzling. It's to make a little bit started to look outside the exactly the same time point, nice, now in the draining lymph node instead of looking in the tumor. And what was evident to us was that there's still a lot of immune cell proliferation happening outside of the tumor, in this case in the lymph node. We saw something similar when we looked in the spleen or in the blood. So what we concluded is that immune cell proliferation is really not happening in the tumor, but it is happening outside of the tumor at these more distal sites or regional sites, including the draining lymph node. And that was interesting to us because at the time, the prevailing model for how immunotherapies worked was to reactivate immune cells that were already present in the tumor. And what our data seemed to be suggesting is that maybe instead we were actually eliciting new immune cells from outside of the tumor to proliferate and then migrate into the tumor to mediate the effective immunotherapy. And so we thought, is there an experiment that we can devise to test that hypothesis, whether we need new immune cells from outside of the tumor? And so to do this, we treated all of our mice with an effective immunotherapy, but in half of our mice, we also gave them a molecule that prevents immune cells from leaving the lymph nodes in the spleen. This is a drug called FTY720 that binds to the sphingosine-1 phosphate receptor, and that's the receptor that immune cells use to sense a gradient of sphingosine-1 phosphate, which leads them outside of the lymph nodes and the spleen to egress from those sites. And so essentially what we're doing here is we're asking the question, if we trap immune cells that are in lymphoid organs there, and we restrict the immune response in the tumor only to the immune cells that were already present in the tumor at the time of immunotherapy, are we still able to reject the tumors? And the answer, as you can see pretty clearly, is no. The mice that got immunotherapy alone, their tumors shrink over time. The mice that got immunotherapy plus FTY720 to prevent egress, the tumors continue to grow over time. And so that seemed to be consistent with our hypothesis that you need new immune cells from outside of the tumor. Now, we thought maybe this is just a consequence of this sort of new immunotherapy that we were developing. Let's go back and look at a model that is responsive to checkpoint inhibitors. Is the same thing true? And in particular, we looked at a model responsive to anti-PD-1 antibodies. The notion really of how anti-PD-1 worked at the time was that anti-PD-1 was interfering with PD-1 expressed by T cells, recognizing PD-L1 expressed by tumor cells or myeloid cells within the tumor microenvironment, and preventing these cells from receiving this inhibitory signal and allowing them to kill target cells instead. So really focused on the tumor microenvironment as the mechanism of action for anti-PD-1. And we were surprised to see exactly the same result that we saw with our other immunotherapy. So here we looked at a mouse model of colorectal cancer called MC38, and we saw exactly the same result. If we left the mice untreated in green, the tumors grow over time. If we treat them with anti-PD-1 alone, the mice control their tumors for this period of 2 weeks. If we give them anti-PD-1 but prevent egress of immune cells from the lymph nodes and the spleen, we see that the tumors continue to grow as if they were untreated. So again, suggesting that anti-PD-1 functions by activating immune cells outside of the tumor that then migrate back into the tumor to mediate their effect. Now, we're not the only ones who think this today, which is great. There are a number of other studies that have come to the same conclusion. And one that I want to highlight, a really nice study by Katie Yost and Ansu Satpathy and Howard Cheng's lab at Stanford, did some beautiful work using single cell genomics to arrive at a similar conclusion. And this is directly in patients. So what Katie and Ansu did was to perform single cell RNA sequencing with paired TCR sequencing from pre-treatment and post-treatment samples of basal cell carcinoma. So these patients received checkpoint inhibitors and they were able to compare the T cell clones that existed in the tumor before therapy and after therapy, as well as the activation states of those cells. And what they found is that the vast majority of cells that were activated after immunotherapy of T cells represented clones that were not present in the tumor before the therapy was given. So these were new clones that were emerging in the tumor after immunotherapy, again, suggestive of this idea that you need new immune cells to come in from outside of the tumor to mediate the effectiveness, in this case, in patients. And so just to summarize what I've shown you so far, what we think now is that systemic immunity, immune cells from outside of the tumor, are required for effective cancer immunotherapy, at least in many contexts. That we've known for a long time that tumors alter immune function within the tumor microenvironment. There's been a lot of work on that topic over the last number of decades. And where we felt that there was still not a whole lot of detailed understanding was the question of whether tumor development alters the systemic immune state. So we know we need immune cells from outside of the tumor for good immunotherapy, but is the tumor doing anything to alter or manipulate the state of these cells outside of the tumor? So that's the question that I will tell you about now that we've been working on over the last few years. And this was work that was led by two fantastic graduate students in the lab, Bree Allen and Kamir Hayam Galvez, who have both recently finished up. And the question that they were asking is, does tumor development alter the systemic immune state? So what they started looking at is, across 7 different mouse models of cancer, how much variability is there in the tumor microenvironment and how much variability is there outside of the tumor? And so here, this is an analysis again of CyTOF data generated across these 7 different mouse models. These represent different types of cancer: breast cancer, melanoma, pancreatic cancer, colorectal cancer, and glioblastoma. And some of these models are transplantable, and some of them are from genetically engineered mouse models. So kind of a wide swath of tumor models that people may use to study tumor immunology. And what we saw looking in the tumor microenvironment was not particularly surprising. There's quite a bit of variability in the total amount of immune cell infiltration, plotted at the top. And within those immune cells, there's quite a lot of variability in the composition of which cells are infiltrating the tumors. And we could summarize that by principal components at the right. And you can see by PCA that each tumor type creates its own unique tumor microenvironment, which leads to these sort of unique islands of color in the plot, where each color represents a mouse from a different tumor model. And what we were surprised to find is that if we looked outside of the tumor, in this case in the spleen or the blood or the lymph node or the bone marrow, we also saw something similar, that each tumor type was changing the systemic immune state in distinct but meaningful ways. And so what we're seeing here is the gray circles represent the cancer-free healthy litter mates, and each colored circle represents a mouse with cancer. And what we're summarizing here by PCA is the abundance of all of the different immune cell types in this particular tissue. And so again, you can see that there's not a whole lot of overlap between the healthy mice in gray and mice with cancer, and each cancer model tends to induce different types of systemic immune alterations. So just as tumors elicit unique microenvironments, here we're seeing that tumors also elicit unique systemic macroenvironments. Now, we focused in specifically on T cells, and we did this because there's been quite a lot of work recently on a process called T cell exhaustion, which takes place when T cells are chronically seeing their antigen. And this is a process that's well described in the tumor microenvironment and originates from an understanding of chronic viral infection. So when T cells continue to see their antigen and are stimulated through the T cell receptor, they undergo this process called T cell exhaustion, where they gradually lose effector functions like making cytokines and proliferating. And because this process is dependent on chronic antigen exposure, we were interested to see whether this process was observable in the tumor uniquely, or whether it was also observable in the periphery. And what you can see, so what we're looking at here is a heat map where the abundance of each T cell cluster identified in our data is colored based on its relative abundance. And we've ordered the data chronologically from left to right. So within each tissue, as you move from left to right, you're looking at the control animals with no tumors, day 7, day 14, day 21, day 35, which is an endpoint for this experiment. The tumors are quite large. This is a mouse model of breast cancer that we're focusing in on here. And what you can see across every tissue is that there are dynamic changes in the T cell compartment, not just within the tumor, but also in these distal sites as well. And that's summarized at the right in this stacked bar graph, where each color represents a different tissue. And we're looking at the fold change here between the earliest tumor size and the latest tumor size to see sort of in summary how these T cells change in composition over the course of tumorigenesis and tumor progression. And I'll draw your attention here to cluster 10, which expresses canonical T cell exhaustion markers. And what you can see is that cluster 10 is pretty uniquely expanding in the tumor and not so much expanding at other distal sites in these animals. And so that led us to the hypothesis that exhausted T cells were unique to the tumor microenvironment, and this process taking place outside of the tumor might represent a different sort of process. And so we asked that question using an orthogonal definition of exhausted T cells. Here are a couple markers identified by Andrea Scheidinger's group at MSK, CD101 and CD38. And you can see using a different definition of exhausted T cells, we saw a similar result, that there are a substantial portion of T cells in the tumor microenvironment that express these exhaustion markers. But if we look outside of the tumor, say in the spleen, we don't really see these cells accumulating. And so the next question we wanted to ask is, do these changes matter? We're seeing these broad changes outside of the tumor. Do they matter for the way that new immune responses take place? And so we took mice with and without cancer, and we asked them to make a new type 1 immune response. So in this case, we infected mice with either LCMV or with Listeria, two model pathogens that both elicit type 1 immune responses. And we asked, do these mice make strong immune responses? And what you can see is the mice with cancer, here shown on the right, AT3 is our mouse model of breast cancer. The extent of T cell proliferation, measured by Ki67 expression, or cytotoxicity, measured by granzyme B expression, was markedly reduced in the mice with cancer. And we can see that quantified at the right. So we're making a much weaker CD8 T cell response if there's already a pre-existing cancer. And this is to a pathogen. Both LCMV and Listeria primarily infect the spleen and the liver, so sites that are far away from the mammary gland where the tumor is. And these also don't express antigens that are shared with the tumor. So really, we're asking the system to make a new response and saying that even these new type 1 immune responses outside of the tumor are systemically impaired. Now, the process of T cell exhaustion has been shown to become permanent through epigenetic modifications that are made in those cells. And so the next question we wanted to ask is this peripheral systemic immune dysfunction permanent? And so to answer that question, we took T cells from either a control or tumor-bearing mice, and we put them in a competitive transfer experiment. So we isolated these cells, we labeled them with fluorescent dyes that get diluted out as the cells proliferate. And then we adaptively transferred them into new control mice that didn't have tumors and infected them with this pathogen Listeria. And we asked, does it matter where the cells come from in terms of their ability to mount a good response? And what we saw was no. The T cells that came from the control mice or the tumor-bearing mice proliferated equivalently. They dilute out the dye from right to left. So you can see they've undergone a similar number of cell divisions, irrespective of whether the cells originated from a healthy mouse or a mouse with cancer, suggesting that this is not a permanent defect that's taking place in these systemic T cells. Now, we also did the experiment the other way around, where we transferred them into tumor-bearing mice at the time of infection. And what you can see here, we're looking at the total number of transferred T cells. So these cells clonally expand when they get activated. And you can see in the gray bars that these cells expand to a similar extent in control mice, regardless of whether they come from control or tumor-bearing donors. So that's the data that I just showed you, represented in a different way. And what you see in the two red bars is that, again, regardless of whether the T cells come from control or tumor-bearing donors, if they're in a tumor-bearing mouse at the time of infection, they do relatively poorly and fail to expand. So again, suggesting that this is not a permanent deficit in T cells, but really it's dependent on the physiological context in which the T cells are being primed. So upstream of T cells are dendritic cells. They're the cells responsible for priming T cell responses. And if this wasn't a permanent impairment in T cells, we thought maybe this is an impairment in the ability of dendritic cells to prime new T cell responses. And so that's the question that we asked. We looked earlier in infection at day 2 when most of the priming takes place. And looking at the dendritic cells, we found that DCs from tumor-bearing mice in the red bar have reduced expression of these key costimulatory and activation markers compared to DCs from the control mice in the gray bar. So after infection, you can see that the DCs from control mice radically upregulate CD80, CD86, and CD83, 80 and 86 being key costimulatory molecules, 83 being an activation marker in DCs. And you can see in the dark red bars that the infected mice with breast cancer have much more weakly activated dendritic cells. And so then we asked, are we able to rescue T cell responses in the periphery if we provide additional activating signals to these dendritic cells? Now, we had previously found that giving a drug called anti-CD40, which activates a co-stimulatory receptor on DCs, can drive strong DC activation, including upregulation of the co-stimulatory molecule CD86. And so we asked, does anti-CD40 rescue these defective systemic T cell responses in mice with cancer? And we also tested a couple perturbations directed at the T cells themselves, like giving them extra IL-12 or giving them anti-CTLA4 to see if we could rescue at the level of the T cell. And what we found is that these T cell directed interventions were not able to rescue these responses, but anti-CD40 was uniquely able to rescue systemic CD8 T cell responses. And so if we walk through the data here in the dark gray bar, we're seeing that control mice that are infected have large numbers of proliferating CD8 T cells. Mice with cancer have a reduction if we look at the dark red bar. In the checkerboard, these are mice that received extra IL-12 and that had tumors, and there's no change there. But if we give them mice with tumors anti-CD40 in the striped bar, we see that we are able to rescue the CD8 T cell response to the infection. Now, because these changes are plastic, something else we wondered is, could we reverse these systemic immune changes by taking out the tumor surgically? And so what we're looking at here are scaffold maps that represent the fold change of all the different clusters of immune cells in the spleen, the site far away from the tumor, in mice that had their tumors resected. And on the left, what you can see is comparing control mice that have never seen a tumor before to mice that had a large, bulky primary tumor that was surgically resected. The systemic state of their immune systems looks very similar to one another, suggesting that surgical resection is able to restore a lot of these systemic immune changes. And what we saw, on the right is comparing the resected mice to mice that still had their tumors intact, that their immune systems looked really different and generally mirrored a lot of the types of changes that we saw comparing control mice to mice with tumors. So again, showing that surgical resection is able to restore a lot of these elements of the systemic immune state, just removing the tumor from the animal. So this does seem to be a very plastic immune state outside of the tumor that is responsive to different types of perturbations, including removing the primary tumor. And here on the right, this is summarized by principal components. You can see that the resected mice have immune states much more similar to the control mice, both at the level of total immune cells, as well as just looking at the composition of T cell clusters at the bottom. And so one of the questions that we asked is, do these mice that have had tumors but that have been surgically removed, have we restored their ability to mount a new immune response to a challenge like listeria? And the answer was yes. You can see if we look at the extent of CD8 T cell proliferation at the top or cytotoxicity at the bottom, that the resected mice perform equivalently to the control mice and much better to the mice that still have their tumors intact. Again, suggesting that these systemic immune alterations are reversible. Now, just in the last minute, I just want to highlight one of the mechanisms that we found that drive some of these systemic changes in this mouse model of breast cancer. So we were looking for soluble factors here that were produced by the tumor cells and that increased in the mice with cancer and decreased after the mice were surgically resected to nominate candidate molecules that could be drivers of these systemic immune alterations. And what we found were two different soluble factors that met those criteria, IL-1 alpha and GCSF. And we also tested TGF-beta, which is a cytokine known to be produced by lots of tumors as somewhat of a control, but also to test whether it might be important in this process. And what we're looking at here is the magnitude of the change in immune cell composition outside of the tumor, whether we administer blocking antibodies to these soluble factors produced by the tumor or not. So we've represented this as compositional distance from the control state. And what you can see again is that tumors induce a large change in the systemic immune state. If we block IL-1 or if we block GCSF, we can partially prevent those systemic immune changes from taking place. And blocking TGF-beta does not have an effect. And if we look specifically at the T cells outside of the tumor, you can see that really IL-1, blocking IL-1, prevents to a certain extent these systemic changes taking place to the T cell compartment. GCSF seems to be dispensable for this T cell reorganization. And if we look here at the abundance of naive CD8 T cells or central memory CD8 T cells, we are able to restore those by blocking IL-1 in this context. So IL-1 seems to be in this setting a driver of these systemic immune alterations, including to T cells. And so with that, I will summarize part 2 here. Hopefully what I've convinced you is that tumor development induces an immune macroenvironment, that the composition of many immune cell types, but also particularly T cells, is altered across the organism during cancer development and progression, that new immune responses are impaired in the context of cancer. And I think the aspect of this that we're excited about and that gives us a lot of hope is that these systemic immune cells remain quite plastic in cancer. So while exhausted T cells may undergo irreversible epigenetic modifications, in the periphery, what we're seeing is a lot of plasticity in the system, and that is quite encouraging, thinking about trying to recruit those cells by using immunotherapies for treating patients. And so with that, I will wrap up. give big acknowledgments to everybody in the group that worked on this project, in particular, Bree and Kamir, who led all of this work, and our collaborators at UCSF, Stanford, Tel Aviv, and the Parker Institute, our funding sources. And I'm very happy to take any questions that you may have. Thanks so much for joining this morning and for your attention.
Thank you, Matt. Rikia, do you want to unmute and moderate? Before we get to the audience question, Rikia, you can moderate. I want to quickly ask one question first. I have so many questions, but I'll say first one. So on the second part, that was very exciting that you showed tumor actually changed systematic immune response. Are there any evidence from human data?
There is some evidence from humans. It's a little bit mixed because most cancer patients get treated as soon as their cancer is identified. And so it's difficult to study what happens in untreated cancer patients, given that fact. And we know that some conventional cancer therapies like chemotherapy and radiation can alter the immune system. And so some of the analyses that exist are a little bit confounded by that a lot of these patients are on treatment. But we do know that cancer patients generally mount weaker immune responses to vaccines. It's harder to vaccinate cancer patients compared to healthy individuals. And there's some interesting data, recently during the COVID pandemic, finding that cancer patients actually have worse outcomes to SARS-CoV-2 infection compared to otherwise healthy individuals of similar demographics. And so yes, there is evidence. I will also say that even in these cases where we're seeing diminished T cell responses in the mice with cancer, those mice are also still able to clear the infection. So it doesn't, it's not as though their immune system is so compromised that they can't clear these very strong stimuli like pathogens. A lot of that clearance is mediated by innate immune cells, and the requirement for T cells is not as strong. And so that could be true in cancer patients as well, and perhaps why we don't see cancer patients coming down with lots of opportunistic infections, at least until they get later in tumor progression. But that doesn't necessarily, just because the immune system is capable of clearing the pathogen, it doesn't mean that the immune response isn't altered in a way that's important for immunotherapy. And so, that's where we're really focusing is how can we take the lessons that we've learned from these studies to develop better immunotherapies that are more efficient at priming systemic T cell responses.
I see. And just, that was on the late stage, but I'm just wondering if it happens early stage enough and the signal's clear enough, maybe it can even augment, like say, liquid biopsy, or maybe that's just too far a stretch.
Yeah, we do see systemic changes taking place at early stages. So even in our mouse models, even after 1 week of immunotherapy, 1 week, after tumor challenge, we do start to see systemic immune changes taking place. So I do think that it's very probable that something like immune monitoring of peripheral blood could be used as a complement to liquid biopsy. The impairment in new T cell responses gradually takes place over time. And so in mice with early tumors, very small tumors, we don't see this impairment in new T cell responses to the pathogen. And we see that sort of gradually take place as the tumors get larger. But the changes in immune cell composition and activation state do start taking place very early as the tumors develop.
Thank you.
Does anyone else have any questions? I'm seeing on chat Eugenio.
Oh yes, should I go ahead and just read them out?
Yeah, sure.
All right, so a question: if you analyze the bone marrow precursor of myeloid dendritic cells, do you see any change in the tumor-bearing mice? That's a great question. We do see a little bit in this mouse model. It's not as dramatic, but it is present. So generally what we see in the bone marrow is a skewing of myeloid progenitors more toward granulocytic progenitors and away from dendritic cell macrophage progenitors. And others have reported that as well. David DiNardo has done some beautiful work at WashU on that topic. And so that is certainly something that is happening. In this particular model, we don't see that mature dendritic cell numbers are impacted. Others have reported that in other systems. We don't necessarily see that in our system. So it's not as though there is a dramatic reduction in the abundance of these dendritic cells. In this system, it seems to be more that the dendritic cells aren't getting activated properly. But in other contexts, people have reported that the numbers of dendritic cells can be decreased as a result of this skewing of myelopoiesis in the bone marrow. And then another question, wondering what would happen if you reconstitute an irradiated animal with bone marrow coming from a tumor-bearing mouse. Do you still have a dampened immune response compared to an animal reconstituted with a control bone marrow? That's a really interesting question. We haven't done that experiment. And it would be really fascinating to do, based on the fact that a lot of the changes we see in the periphery seem to be dependent on continual, the continual presence of the tumor. If we resect the tumor, it goes back to normal. My suspicion is if we reconstituted control mice that we would get similar immune cell composition, but we have not actually investigated that, and it would be really interesting to do so. So thank you for the suggestion.
Thanks a lot for answering. Anyone else has a question? You can write it here or you can raise your hand up. Or just speak. Okay.
All right. Well, if there are any other questions that folks come up with, feel free to reach out. Always happy to answer emails. So feel free to send me a note.
Thanks a lot, Dr. Spitzer. Professor Huang, do we have any other question?
I think we're good. I thought I saw someone raise a hand, but maybe that was just no. Okay. Sounds. Oh yeah, I think Jesus have a question. You can unmute yourself and ask. Feel free.
Thank you so much for this awesome presentation. I am thinking about this model of human tumors, these so-called immune phenotypes. Well, they see that highly infiltrated tumors by T cells correlate with better immunotherapy response and, in general, better higher survival. And I'm thinking if this correlation that we see, they see in human tumors, is not because the tumors are highly infiltrated, but they allow infiltration of newly activated T cells, as you see in your models.
Yes, I think that is very likely to be the case, that the higher infiltration in some human tumors is reflective of what's happening outside of the tumor as well. And there's some really nice data soon from some T cells in the lymph node, a particular subset of antigen-specific T cells in the lymph node that seem to be responsible for producing new T cells that infiltrate into the tumor, not in the context of immunotherapy, but just sort of steady state responses to tumor progression and initiation. And so I do think that that's likely to be the case, that if you're seeing more T cells in the tumor, it's also reflective of there being a stronger T cell response to the tumor outside in lymphoid organs, and that those cells could be, really the drivers of immunotherapy responses.
And just to follow up a question on the other side of that, tumors with some genotypes are known to have immune evasion. Do you think that reflects a systematic change as well? Or, people previously think that there's just no immune cells in the tumor for the immunotherapy to be effective, but maybe it's actually the systematic effect.
Yeah, that's a great question. And I think that some element of that is likely to be the case. There are probably some escape mutations that tumors can create that are more local in terms of their impact. Things like losing MHC class 1 expression or beta 2 microglobulin, or mutations in the JAK-STAT signaling pathway, those types of mutations may allow a tumor to escape even if there is a local T cell response that could be mounted against it. But in these cold tumors that just don't have much T cell infiltration to begin with, I think it is likely that those are also reflective of sort of poor priming to those tumors in the periphery, and that might really be the driver of why there aren't T cells in the tumor, is that there's just no real immune response taking place outside of the tumor as well.
Thanks.
But all really great questions that clearly we need to do more work to understand in more detail, especially in patients.
Yeah.
Thanks a lot. This was an amazing presentation. And if no one else has a question, we will wrap up our session here. Is that okay with you, Professor Huang?
Yeah, sounds good.
Yeah.
Thank you so much, Matt, for an awesome presentation. Thanks everyone for your questions and listening.
My pleasure. Thanks for the invitation.
See you guys. Bye.